Is speaking to scientists. It's about scientists sharing their work with the lay public, and even our communication participants, sorry, our communication colleagues. If you're working on a communication initiative, you can always use this platform. Use simple English. Imagine you're speaking to a primary 7 or grade 7 child. That is the kind of communication that we want, for you to step down your research and make sure that, you know, the general public can understand. And this is not limited to only Nairobi. We would like this to be an IITA thing and eventually maybe a CG thing. And we are going to, as the presentation comes to an end, we shall send out a poll and ask everyone to score Trushar on those parameters that I've talked about. Was he simple? Was he understandable? Was he conversational? Was it fun? You know, because that's what now we want to bring into our research circles: fun, conversational, something you can remember and relate to. And at the end of the year, we shall score all our presenters, and the Alliance for Science, which is a co-host in this webinar series, will give out an extremely beautiful and important and yet very small gift to the winner of this webinar series. So this is really giving our scientists the opportunity to communicate their science in a very simple, fun, and conversational way. Just to talk a little bit about the Alliance for Science, who are our co-hosts in tandem with IITA Nairobi. The Alliance for Science is a communication initiative, and we are headquartered here in Nairobi, and Trushar is our host. So this is what we do. We want to take science to the people. We want science to be uptaken, consumed, and adopted. So today we are very honored to have Trushar as our first speaker. I'm going to stop sharing, and I'll ask Trushar to introduce himself first before he can go into his presentation. Thank you. Over to you, Trushar.
Okay, thank you, Patricia, and thanks for this opportunity. It's been, I should say, one of the hardest things I've done, just thinking about it. There's two things that are terrifying me. One is presenting as a scientist in language that standard 3 or standard 7 would understand. And the other thing that terrifies me is thinking of you all in uniform as standard 3 students. So, okay. My name is Trushar Shah. As Patricia has said, I work as a bioinformatician, and hopefully by the end of this seminar or this webinar, you will know what I do. What does bioinformatics mean? How does it apply to farmers? How does it apply to people who are working in the area of agriculture? And what is it all about? Okay, so since we're all in class, I thought the first thing we will do is we'll do a bit of an exercise. Okay, so this is your first practical exercise as the classroom here. And also people online, please feel free to participate. I just want you to follow or enact or do whatever appears on the screen. Okay, so people in the room here. Thank you. Thanks for that applause. Next one. Okay, another one. Wow. Another one. Okay, so we lost some people. Another one. Wow, we lost a few more people. Okay. I know Patricia mentioned that this should be in English, but I'll give you the why we're going through this exercise. Now, you know, in Kenya, especially, we mix up our L's and our R's, isn't it? So I actually want you to do the actions that you did before, but on screen, something else will appear. Okay, so what happened? Okay, what's the moral behind all of this exercise that we did? So we had basically a set of instructions. Okay, we had an alphabet. Okay, the 4 letters that you saw consist of an arrangement of 26 letters in a specific order, and that gives you an instruction. When I said clap once, it gave you a kind of measure. Rather than clapping many times, it told you clap only once. The piga makofi and the tuhure moko was doing the same action, but using a different language. So for those of you who don't know, the piga makofi is Swahili, and the tuhure moko is from the Mount Kenya region. Okay, and one of the biggest tribal groups in Kenya. So it's the language coming from the Kikuyus. Okay, and what happened when I was typing clap on my computer, instead of typing clap, the L changed to an R and I got a typo. And this typo is what we call a mutation or a variation. So suddenly the instruction to clap became something very different. Okay, I hope I haven't lost you all. So similarly, in life, in living things, we also have a language, okay? And the language of life is something called DNA, deoxyribonucleic acid. That is basically the alphabet. In this case, the alphabet consists of only 4 letters, not 26. And you can see here in this cartoon over here, that alphabet, the 4 letters of that alphabet are A, C, G, and T. Okay? So think of those as the letters. Okay? The arrangement of those letters gives you some kind of instruction. Okay? So this is DNA, is the building blocks of those letters, the alphabet. This next thing here on the right, I don't know if you can see my cursor, but the sort of blue squiggly diagram is what we call RNA. So think of that as the language of instruction, okay? So the first DNA is the arrangement of letters. The second is the language of instruction, okay? And the third is the protein, which is the action. So the action of clapping, the action of building something, the hands coming together. You need the hands to be able to clap. When I said clap, you didn't clap with your feet, right? Because you understand that language. Okay. So this is the language of living organisms. And this living organisms basically represents the bio in bioinformatics. So it's the bio means life and the study of living organisms. So we're looking at living organisms here, and that could be anything from a virus to a human to a plant, anything that is made up of cells and has either DNA or RNA in it, okay, as the language. Okay, so when we talk about genomes, let's think of the DNA as the alphabet, but the genome is what constitutes the entire set of instructions for life, okay? So it's like your manual for life, if you want to call it that. Okay, and you can see here genomes come in various sizes. So you can see we have genomes of viruses right up to genomes of flowering plants, and there's a variation down here. It shows you the size. And because I'm a scientist, I like to use these, you know, instead of just calling it as 1 million, I like to call it 10 with 6 zeros following. So I call it 10 to the power 6, okay? So that means if we look at the human genome here, the mammals, the size of the human genome constitutes of 3 billion letters, 3 billion bases. The A, C's, G's, and T's are called bases. Okay. So this, if I was to give you a set of instructions and I was to print out A, C, G, and T in different order, okay, as a set of instructions, I was to print it out, I was to put it on a Word document, if Word could handle it, and print it out in reams of paper. Just to give you a vision of the magnitude of it, if I pile those papers up, the printed papers with the whole human genetic code, it would be, if I piled up from the bottom road, it would be all the way up to the Nairobi Expressway. That is the amount of paper that I would consume just to see. And this is the genetic, this is the instructions that are actually in your cells, in the nucleus of your cells, okay? Which is the building blocks of your organism. Okay, so that's a lot of data. Now, if I was to tell you, find me the instruction clap in this ream of papers that goes from the bottom of the road right to the top of the expressway, and you didn't have Google, it would be a very tough task, right? So our jobs as bioinformaticians is to then become the Google and the IKEA of decoding this set of instructions. Okay, and I'll explain that in a minute. Let me just take you a little bit through genome sequencing. And if I just take you through, just look at this amazing advancement in technology. If in 2001, which is, 20 years ago, I wanted to sequence the human genome, it would have cost me approximately $100 million. Okay. Today, I can do it in under $100. And all it involves is someone sending you a kit. We're all familiar with kits now, especially with this whole COVID pandemic. You get a kit, you take a swab of your saliva, you send it back, and they will give you your entire genome sequence, okay, for about $100. There are services out there that exist that can do that. So just to tell you that the cost of the sequencing has come down a lot. But what has that done? It has exploded the amount of data that is now available, because rather than sequencing one representative of a genome or a species, rather than sequencing one maize plant to represent all maize plants, I can now go to individual plants and start sequencing them. Okay, so start looking at how these are coded. Okay, so you can imagine if I do 3 billion times the number of different maize type of plants that I have, that's a lot of data and information. So that data and information is now handled through what we're calling bioinformatics. Okay, so the informatics is the computational part of it. I need tools, I need to be able to store this data. I'm not going to print it out and put it in papers. I need to be able to store it. So we need to have the right formats to store it. We need to have the right amount of space to store it and all of that. To do that, we need high computational power. I'm not going to be able to open such a big file on my cell phone. Even though cell phones have enough power, the same amount of power that the first computers that sent people to the moon have. But to open such a file or to open such a set of instructions, I would need very high computational power. The tools I would need to figure out which language am I talking in? how do I look for these words in these languages? Okay. And also I need, for example, I already know what clap means. Clap in English means putting my hands together, but clap in another language could mean something else. Okay. So providing the references, basically the dictionaries and the thesaurus and the translation of all these biological languages that we speak. A good way to mine through this data is also to visualize it. You know, some of the things that I'm presenting, if you see it visually, it evokes more of a response to say, wow, look at this graph here. Something happened in this year over here around 2009, 2010, or 2007, where we see this dramatic decrease in the cost of sequencing. And so what happened then was there were new technologies developed called next generation sequencing. This was the first generation of sequencing technologies. And suddenly the price and the amount of data, the price came down, the amount of data went up. And that's so because I present this in a visual way, I'm able to look at trends. I'm able to compare it to other sets of information. So we provide tools that allow us to visualize, and obviously the internet to connect to other existing databases. Okay, so what I was saying here in a nutshell is bioinformatics is made up of two parts: the biology, which is the DNA sequence, the RNA sequence, the protein sequence or alphabet, as we want to call it, and the informatics, the tools, the infrastructure, the kind of hardware, you know, in terms of the roads, the hospitals, all of that we require to be able to store that information, to be able to mine that information. Okay. And so we generate data from biology, and we then use computational tools to interpret that data and give a set of results. Okay, so that is in a nutshell what bioinformatics is. In generating all of this, we generate a lot of data repositories. It's like Google becoming a store. By you publishing something on the web, it automatically gets mined by Google. Okay, whether it's a newspaper, whether it's a scientific protocol, whether it's a video, everything gets the repository then becomes mined by Google. Similarly, we as bioinformaticians provide tools to be able to mine biological data. Okay, and then generate knowledge from there. So I think Patricia asked me one question in the end. She said, why should a farmer care about bioinformatics? What is in bioinformatics that a farmer should care? So remember, farmers would be interested, for example, if I was a banana farmer, I would be interested, depending on which region I came from, in a specific type of banana. I might want one for cooking with a specific taste, which has a specific size, and which matures in my farm very quickly. Okay. So that's something that I may be interested in. And this is what we call the diversity. So we can analyze the diversity of different crops. Okay. As a farmer, I may be affected by disease. Over here, you can see some kind of disease may affect me. So I want to know more about the disease. If it's a new disease that's coming, we have many occurrences of that. We've just been through a pandemic, and you already know, I can ask you, okay, you know coronavirus, and then you know the variants of corona. You know Omicron, and you know Delta, at least, right? So those are possible because of a combination of biology, molecular biology, so the kind of things that Valentine, Lena, Jay, and them do in their group, and bioinformatics, which allows you to then mine that information. Okay, so we want to look at pests and diseases, and so we can start decoding the genomes of the varieties which are resistant or susceptible, and see what is the instruction, what is that in the language of the DNA that is the instruction that says, I am resistant or I am susceptible to a particular disease. We can also start looking at the instructions of the pathogen to say, what makes this pathogen attack bananas and not sorghum? Okay. What is in the molecular mechanism that makes me prefer bananas over sorghum? So we can start looking at that because we can decode the DNA of the pathogen itself. And then finally, as a farmer, I may say, I like this one, but it doesn't grow well in my farm. So can I have a close relative of this? Right? And so I would want to then go to the database or the gene bank in this case and say, okay, I have something that meets your criteria. You wanted a banana that looks like this, that tastes like this, that grows like this, and is adaptable to my environment. Okay, so I can grow it nicely in my farm here in Uthiru or here in Kikuyu or in Kisii or whatever I want to grow it. Okay, so that would be about the adaptation. So we can, through bioinformatics, help with mining the diversity, looking at pests and diseases and the responses, and looking at how specific characteristics or traits, as we call them, are linked to a particular genotype, which means are linked to a particular DNA code of that specific plant or organism. Okay. So that's what I have until now. I think in future seminars, we will continue with the work that, for example, this work, if I say, here is a language of instruction that I have decoded, I can pass that on to someone else like Valentine or Lena or Sam or Mark, and they would be able to then include that set of instructions in a plant so that it becomes taller, it becomes more resistant to disease, it becomes higher yielding, things like that. So they can then go in, work in the lab, do their reactions, and create a plant that will hopefully understand that language of instruction and interpret it to produce an action. Okay, so that's where I'm going to leave it. I was told 10 minutes, so hopefully I haven't confused you totally, and I've given you an insight into what we're trying to do in terms of finding needles in haystacks. We want to find those signals in the large ream of papers that we have with a set of instructions.
Thank you. Thank you, Trushar. If you could for a moment stop sharing. So Trushar, thank you very much. You know, when you're listening to music, usually somewhere along the 3 to 6 minutes, the musician often refers back to the name of the song, or when you watch a movie, someone throws a one-liner and they mention the movie. You promised you're going to talk about finding, let me find your topic that you promised us. Finding needles, okay, in haystacks. And out of 100, you've already lost 10 points because you used the staking papers up to, you know, the Nairobi highway. Then that should have been your title.
Okay.
But other than that, I think you've done a great job of showing us how you're finding language in that, you know, in that pile of paper and how high it is. So we would like to open up for questions, maybe just 10 to 15 minutes. And we are not going to task our participants to use simple language, but we are going to task Trushar to respond to whatever nature of the question comes in the simplest language possible. So we'll take only 5 questions, as we also send out a link to our poll we request that you please fill it out so that we can score Trushar and see if he's going to get 80 out of 90, since he's now no longer at 100. So please send your questions through chat.
Okay, I just want to justify why I put that title, because the expressway wasn't there a while back, and we're not engineers, we're agriculturists, so haystacks are closer to agriculture than expressways.
Oh, okay. Trushar, it's okay. Thank you very much. Good presentation, excellent presentation with good examples. That is what's coming out through the chat. But we would like for you to ask at least 5 questions, especially people who are in the bioinformatics area.
I have a question.
Yes, you turn on your speaker.
It's on. Hi, everyone. For the sake of those not in the room, my name is Juliet, also based in Nairobi. So I'm one of the laymen. I hope I have understood you. I think I have understood you. Of course, I wish there was time for more. I wonder what would make you study something like this? Like, what is the basis of your interest on a personal level? And then, do the same rules apply between plants and humans, or are there variations?
Okay, so both very good questions. So on a personal level, I would say I've always been fascinated by biology, how things work in living organisms, but I wasn't made up for the lab, right? I didn't have the patience of going, mixing things, waiting for a while, looking at, I wanted, as I moved along, I also developed an interest in computing. And that's where the two came together, the biology side of it, and how I could use computational tools to get a better understanding of the biology or decoding the biology. So that's my personal story. The second question about, do the same rules apply for plants and animals, or plants and humans? Okay. The underlying code is the same. So if you look at the alphabet, it's the same. The same 4 letters are there. But what differs is the language, so the RNA, and what else differs is the protein, because for plants, for example, they need to produce chlorophyll that needs to photosynthesize. They need to make their food from the sunlight. Humans ingest their food and need to process it in a different way. So those instructions will be slightly different. And you could see, because the way things evolve as well, we didn't talk about the evolutionary aspect, which means what are the ancestors? How did they come from? At some point, plants came from, photosynthetic organisms, which means they could produce this chlorophyll, they could survive in sunlight. And so I would say the alphabet is the same, which makes a lot of the tools applicable, right? But they need tweaking in terms of the language and the instructions.
Thank you.
Thank you, Trushar. I can see 12 people have already responded to our link scoring Trushar. Thank you very much. We are quite a number in the room, so I'm sure we'll take time and score him. There is a comment here, but I'm going to turn it into a question. Someone is saying staff should be asked to explain the difference between bioinformatics and computational. No, there was, let me find that. And biometrics.
It's biometrics,
yes.
Yeah.
So he was saying you did that very well, but I'm requesting that if you could explain that difference again.
Okay, sure.
Yes.
So bioinformatics, as I mentioned, is dealing with large amount of data and information coming from living organisms. Okay. Now, when you want to start analyzing that data, or if, for example, I want to start asking questions, I need to then have some kind of statistics. For example, when I was asking everyone to clap, right? If no one in the room knew English, they would have only responded to the first instruction, which was the emoji, right? For those who knew English, they still clapped. For those who knew Swahili, they clapped. And those who knew Kikuyu, they clapped, okay? But the numbers get smaller and smaller. So the biometric side of things deals with those numbers. What is the probability that this instruction is correct? If I'm targeting a group of standard 7 students here, which is the correct language to speak to them in? Okay. And can I get a measure of confidence? And that's where the statistics, the biometrics, the sampling, who is in the room becomes important. Okay. So the biometrics deals with the statistical nature of things, whereas the bioinformatics deals more with the large volumes of data, using tools to mine that data, store that data, analyze that data. So there is gray areas. There's always overlaps, you know. Someone like Valentine or Mark or Sam, they would be doing bioinformatics. It's not that only a bioinformatician can do it, but bioinformaticians have developed tools which allow molecular biologists to mine that data, or the person has adapted by learning some programming or learning a particular language to mine that data. So that's the difference.
Thank you, Trushar. We'll take only 2 more questions. I thank you for your interest. Oh, sorry, thank you. So I was saying we'll take only maybe 2 more questions, and we thank our online audience for your interest, and of course our in-person audience here. So Chamba is saying, thank you, Trushar, for the nice presentation. How can bioinformatics be used to develop gene bank collection subsets useful to breeding programs? And Trushar, part of simplicity is brevity. Thank you.
Okay. So then I will just maybe explain that visually, since you would like me to be brief. I touched on that. So in the gene bank in Ibadan, we have bananas, plantains of all different types stored over there. If I was to grow it in the field, I could get a banana that looks like this, or I could get a banana that tastes sweet like that. So when the breeding programs develop products, and by products we mean specific characteristics are included in that product, which is the banana plant, which will produce a certain type of banana. For example, one that cooks well, one that produces a big bunch, that's the yield, one that grows fast, you know, early maturing. We can then go and mine all of that data and start creating that these are the subsets. If I wanted something that was red in color, I would use this and its relatives as a subset from the gene bank to introduce that red color into the banana skin, if that was appealing for anything. So that's how we would use the molecular information. So basically the DNA sequencing of all these different varieties in the gene bank, we could then link that to what is the specific traits or the phenotypes, which we call. So what is the characteristics that that subset represents?
Okay. Pinihas is saying, very interesting presentation. Elaborate how you will give a farmer a variety that is doing well in her environment, although Pinihas said is using biometrics.
Okay. So the idea here is, I wouldn't use only biometrics, okay? I would give the language to someone like Valentine. I would say, this is the instruction you need to give that particular plant, okay? The instruction is grow short, but produce a big bunch. So if I know the verbs for that from the 4-letter language, I can give that to someone like Valentine, and he can then edit the language so that if it was crap, it becomes clap. Yeah. So those variations can be produced. And then obviously he would test that the L changing to R has no other effects. It may become short and produce a big bunch, but the taste may be quite bad. So then obviously I would pass it on to Valentine. Valentine would pass it on to the breeders to grow out, to test in the field, whether that, and then from the breeders, it would then go to the farmers, and ultimately they would be the ones saying, yes, this meets the criteria that I wanted, or it's missing X, Y, or Z.
Thank you, Trushar. Finney has, my speaker was off when I read out your question, but that response was to your question. And Trushar has just told us that his work is not directly with the farmer. He passes on the instruction to other people along the pipeline, like the gene editing people here, Valentine and Lina, and they'll do what the farmer wants. And there are several other people along the research pipeline. Yes, Finney has, thank you. And because Trushar has gone above and beyond the call of duty to explain gene editing and what happens, I give him back his 10 marks. So we are marking him out of 100. And we are taking our final question from Solomon, who is saying, did you say kilobase for mammals is less than those of flowering plants? If yes, why?
Okay, that's a great question, Solomon. So yes, I can't answer your question because I don't know why. So when we speak a language, if I was to use English, I said one action word in 4 letters. If I was to use Swahili, I used 2 words, one of 4 letters and one of 6 letters. Okay. So what happens there? The instruction for that was given in English is succinct, direct, to the point, but is only applicable for English speakers, right? The one for Swahili is only applicable for Swahili speakers. Now, it could be, you could be a native, it could be in your genetic code, it could be in your language, or it could be something that you as a person have learned, okay? So the size of that language does not necessarily relate to the size of the organism or the size of the level of instructions, because DNA, we call it, there's a coding part, which means an instructional part, but there's also a non-coding part, which is basically telling you the amounts, the targets. So like we said, clap once. So the once is encoded in another part of the instruction. Okay. If I said once clap, it wouldn't make that much sense, right? As clap once, the way it's arranged. So the once is used in conjunction with the verb, okay, to give it a measure. And so we have within the genome, which is the set of instructions, we have different parts which will form the verb, the noun, the adverb, if you want to think of it as a language. Okay, and those languages can differ in the size, but they have no relation to the complexity of that language group or the ethnic group or whatever it evolved from. So I hope that clarifies that it's just about the language.
Thank you, Trushar. We have 21 people who have voted. We say thank you very much. And this is how we scored him. Was Trushar's presentation easy to understand? 4.7. Obviously, 99% people in the room are scientists. Could you understand who the beneficiary of his work is? They're saying 4.6. I wish I knew who you are so that I ask you who this beneficiary is.
So these are out of?
5.
Oh, okay.
Yes.
The measure matters. I thought I was
know.
failing.
Thank you. It's not out of 100, it's out of 5. Was it fun and conversational? They are saying 4.6. That is what democracy and elections does. So Trushar looks like he's around 80, 85%. So for everyone who has joined us today online and in person, thank you very much. From all of us in Nairobi, Kenya, we are saying bye-bye, thank you. See you next month. Yes, kwaheri, they are saying. Thank you,
Asante.
bye.
Thank you.